Hi, today we're going to answer the questions: what is secondary hemostasis? What is the extrinsic pathway? What is the intrinsic pathway? What happens when any one of the steps involved in the two pathways goes wrong? What is an anticoagulant and what is an anti-clotting drug? We'll give examples of these drugs and also give you clinical examples of both. At the end of the section, we'll provide you with MCQ questions that will assist in you consolidating the knowledge for today's segment. Hello everyone, my name is Harmony and I'll be a personal physiologist from today onwards. Okay, let's get to it. So, after the end of this video, one should be able to explain briefly what primary hemostasis is. So, if you haven't watched the previous videos, I propose that you go into the channel Doctor of Medicine and look for the first segment in hemostasis, so it will explain broadly what primary hemostasis is. So before I talk about many things, I will give a recap on primary hemostasis. We'll also talk about the involvement of procoagulants in secondary hemostasis. Okay, we'll also mention a lot about the differences between the extrinsic and the intrinsic pathway, and also talk about the link between the two pathways, what we call the common pathway. All right, we'll also explain the natural anti-clotting system, what happens within your body that allows the other parts of your blood vessels to not clot. All right, we'll also talk about anticoagulants and synthetic coagulants, so the drugs that are administered to people to avoid clotting. Lastly, we'll also mention the abnormalities of the process of hemostasis, specifically secondary hemostasis. All right, so just a recap of the procedure or the process that we call primary hemostasis. All right, so a question that comes to mind is, what is hemostasis? As mentioned in the previous segment, hemostasis refers to the stopping of the loss of blood. Okay, so we are stopping the loss of blood. That's hemostasis. Hemo means blood, stasis means to stop. So we want to stop the leaking out of blood in case of an injury. All right, so hemostasis is divided into two. We have primary hemostasis, we have secondary hemostasis. So primary hemostasis refers to the formation of a platelet plug. All right, so at the site of injury, when blood is exposed to the underlying connective tissue, which consists of collagen fibers, there is activation of platelets, causing them to release certain substances, what are called the granules within the platelets, which initiate primary hemostasis. All right, so the diagram in front of you shows a compilation of steps involved in primary hemostasis. All right, so primary hemostasis is divided into three stages. We have platelet adhesion, we have platelet activation, and lastly there is platelet aggregation. All right, so platelet adhesion refers to the binding of platelets to the underlying collagen fibers in the connective tissue. All right, so we need an intermediary factor known as the von Willebrand factor, which allows the platelets to bind to the underlying collagen fibers. So von Willebrand factor may be viewed as a sort of glue, if you may like. It acts as a glue between the two sides, the collagen fibers in the connective tissue and the platelets within the bloodstream. Okay, so we need von Willebrand factor. So when the platelets bind to the collagen fibers, they become activated, they release the granules within them. Most importantly, they release serotonin, known as 5-hydroxy tryptophan. Okay, so serotonin is released from the platelets. We also release adenosine diphosphate, thus ADP. So this is step one. Platelets, they bind to the collagen fibers, they secrete their granules. Thus your step two. We release an enzyme or a protein known as thromboxane A2. So thromboxane will act to further stimulate the activation and aggregation of platelets. The last stage is stage 3. This is your stage 3. So look at this. Down here in the connective tissue, we have your collagen fibers. The ones in red, this is your von Willebrand factor. So the von Willebrand factor, its use is to connect your different platelets, right? So we have new platelets which come and overlap and overlie on top of the old platelets. We call this aggregation of platelets, and it's achieved by your von Willebrand factor. Communication of platelets with the collagen fibers is also achieved by the von Willebrand factor. So we start with platelet adhesion. We secrete the granules that are going to further activate the platelets. So that's going to be referred to as platelet activation. Then lastly, we have platelet aggregation. So now we form a platelet plug. So aggregation of platelets at the site of injury results in formation of the platelet plug. Okay, so clotting or formation of the platelet plug is only confined to the site of injury. Thromboxane will promote aggregation of platelets at the site of injury. Okay, so we have cells on the periphery, on the sides of the blood vessels, which are still intact. We call them endothelial cells. As you can see, the cells which line the inner lumen of your blood vessels. Endothelial cells will secrete or produce nitrogen monoxide and another protein which is similar to thromboxane, referred to as prostacyclin or prostaglandin. Okay, so the prostaglandins will act to inhibit the formation of a platelet plug on the parts of the blood vessels which are intact. So this culminates the process of formation of a platelet plug. So this is where platelet plug formation terminates. Okay, so the next stage that's important in hemostasis is formation of a fibrin meshwork, so which is going to act as a glue on top of the platelet plug. So that's the topic for today, secondary hemostasis. All right, so now secondary hemostasis, let's look at it in detail. So the blood coagulates after addition of exogenous biological materials. This is an observation that was made by researchers back in the day. They realized that if you add some exogenous materials like macerated brain extracts, blood will spontaneously coagulate. Okay, so blood also coagulates when it comes to contact with surfaces like glass. All right, so these two observations were key in the formulation of the two pathways that are involved in the clotting of blood, which we call the intrinsic and the extrinsic or pathways. Right, so the intrinsic pathway is going to be different from the extrinsic pathway, but both pathways are going to involve a series or a group of clotting factors. Clotting factors are going to be inactive plasma proteins, which when activated will be converted into a proteolytic enzyme that will further activate another inactive plasma protein into a proteolytic enzyme. At the end of the day, the cascade will result in the conversion of a soluble protein that is called fibrinogen into its insoluble form, that's fibrin, which is going to form the fibrin meshwork. Okay, so that's what essentially we have in these pathways. So the intrinsic pathway is going to be initiated by contact activation of factor 12, which is going to be exposed to the collagen fibers or glass surfaces, which results in its activation. All right, so if we have 12 and 12A, factor 12 is inactive 12, then 12A is activated 12. All right, so we'll look at as well as the extrinsic pathway, which is activated by a clotting factor known as clotting factor 3, which is otherwise known as thromboplastin or factor 3, right, which will then result in the extrinsic pathway. So blood coagulation involves the following. So the clotting mechanisms will involve a cascade of reactions in which clotting factors are activated. So we have 13 clotting factors that are used in both the two pathways, the intrinsic pathway and the extrinsic pathway. Okay, so most of these plasma proteins are synthesized in the liver by action of vitamin K. We'll look at how the liver is involved in these clotting factors. When activated, these clotting factors will act as proteolytic enzyme, and several of the steps in the coagulation cascade, they require calcium as well as what we call platelet phospholipids. So calcium is known as clotting factor 4. This is a summarization of the different clotting factors that we require in the coagulation cascade. So the coagulation factors have different names, okay? So we call them factors 1 through 13, but they have different names according to the people who discovered them. So factor 1, inactivated factor 1 is known as fibrinogen. Activated factor 1 is called fibrin, okay? Factor 2 is called prothrombin. Activated factor 2 is called thrombin. We have calcium, which is your factor 4. Tissue factor, it's factor 3, is also known as tissue thromboplastin. We have another thing called plasma thromboplastin, which is going to be your factor 11, okay? Plasma thromboplastin antecedent, okay? So the important factors that we need to know their names is going to be fibrinogen, that's your factor 1, prothrombin factor 2. We need to know tissue factor, which is factor 3. Most importantly, we need to know your factor 10. It's called the Stewart power factor. We need to know your factor 13, called the fibrin stabilizing factor. We also need to understand the functions of factor 8, known as the anti-hemophilic factor A. Okay, so let's look at the different coagulation cascades. Now, the intrinsic pathway is initiated by what we call contact activation, as indicated in the diagram above. So contact activation results in the activation of factor 12. Factor 12, also known as the glass factor, is converted to its activated form, which is a proteolytic enzyme, which catalyzes the conversion of factor 11 to factor 11A. Factor 11A will further catalyze the conversion of factor 9 into 9A. Now, the action of factor 9A requires a cofactor, which is your factor 8. So you realize that some of the clotting factors do not act as proteolytic enzymes, but they act as cofactors to the proteolytic enzymes involved in the coagulation cascade, okay? So we have factor 9A with its cofactor, which is factor 8. So factor 8 together with factor 9, they're going to convert factor 10 into factor 10A. Factor 10, known as the Stewart power factor. So the Stewart power factor will then proceed to converting prothrombin into thrombin. Prothrombin is our factor 2, thrombin is the activated factor 2. Thrombin will then proceed into converting factor 1 into factor 1A. We'll see a detailed outline in the next slide, okay? So on the other side, we have the extrinsic pathway. Extrinsic pathway is a bit different from the intrinsic pathway. It's activated by an extrinsic protein known as the tissue factor, otherwise known as thromboplastin, okay? So thromboplastin is found on the cell surface membrane of cells outside your blood circulation. So cells like fibroblasts and other cells outside of your blood circulation. So when the blood comes into contact with these cells, thromboplastin is released. So that's our factor 3. Thromboplastin will convert factor 7 into activated factor 7A. Okay, so your 7A will then split into two pathways. The other pathway provides a link between the extrinsic pathway and the intrinsic pathway. So 7A will activate 9 into 9A. Then 9A will then work together with its cofactor, that is 8A, to form your activated 10A. The other pathway involves the activation of the Stewart power factor by means of your factor 7A. Okay, so there is your 7A goes down, catalyzes the conversion of factor 10 into factor 10A, which then activates prothrombin. Okay, so this is just an introduction to the two pathways. Okay, so let's look at a detailed outline of the two pathways, including the major plasma proteins that are required for the different reactions to take place. So here outlined is a detailed explanation or a detailed view of the two pathways. So I'll start by explaining the intrinsic system. Okay, so the intrinsic system, we say it is activated by contact activation. So when your factor 12 in vivo comes into contact with the underlying collagen fibers, will be activated to its activated form, that's 12A. This happens also in the presence of two important plasma proteins, that is kininogen and kallikrein. Okay, so kallikrein and kininogens are required. So there we go. Factor 12A will then activate the conversion of factor 11 into factor 11A. So we need our high molecular weight kininogen to convert factor 11 into its active form. Factor 11 will activate factor 9. Okay, so in the activation of factor 9 in the intrinsic system, we have that is your factor 11A. Then factor 9 will then move forward, proceed to activating factor 10, the Stewart power factor. For factor 9, we need a cofactor, which is your activated factor 8. Okay, so the activated factor 8 works together with factor 9 and other factors. Here we have our calcium ions, we call calcium factor 4. Okay, we also have the PL, what we call the platelet phospholipids. Okay, so the platelet phospholipids, they will also be involved. These are activated platelets in the activation of factor 10. Then factor 10 will also require the presence of factor 4, which is calcium, platelet phospholipids, and activated factor 5 in order for it to convert prothrombin to thrombin. So I usually say factor 9 requires factor 8 because 9 and 8 are next to one another. Okay, so remember that 9 requires 8. Factor 10 requires factor 5 as its cofactor. These two, they love each other because they are both multiples of 5. Okay, we also need factor 4, which is calcium. Prothrombin is the precursor to thrombin, so it's the inactive form of thrombin. Then thrombin will be converted, will catalyze the conversion of fibrinogen into its insoluble form, that is fibrin. So the fibrin that we produce by use of activated factor 2, which is thrombin, is said to be loose. Okay, so we have loose fibrin strands. We need to stabilize the fibrin strands by establishing cross linkages in between the strands in order for them to form a fibrin meshwork that we are going to superimpose on top of the platelet plug. This step is catalyzed by factor 13. Okay, before factor 13 is able to carry out this stabilization step, it has to be activated by thrombin. Okay, so factor 2 will activate factor 13 into its activated form. Factor 13 is also known as the fibrin stabilizing factor. It then proceeds to stabilize the fibrin, so we produce stabilized fibrin. So this marks the end of the coagulation cascade. So the extrinsic system on the other side initiates with the tissue thromboplastin. Okay, so the tissue factor or the tissue thromboplastin is the initial activator or the initiator of the extrinsic system. So your tissue factor is going to activate factor 7. Factor 7 works in two ways. Either it activates factor 9 or it goes straight to the activation of factor 10. Then from here onwards, from factor 10 going down, we call this the common pathway. Okay, this is the part of the coagulation cascade that is common between the two systems. So the common pathway is outlined here, still the same thing, starting from factor 10, going all the way to the formation of stabilized fibrin by factor 11. So another thing that we really need to mention is this. So thrombin lies at a central position in the coagulation cascade. Thrombin has major functions. One of the functions of thrombin is conversion of fibrinogen into fibrin. The second one is the activation of factor theta. The last function of thrombin is the activation of platelets, okay, as well as providing a positive feedback effect, which goes back to activate factor 5, factors 9, as well as factor 11. So it's factor 5, sorry, factor 8, and factor 11. That's the positive feedback scheme. All right. So the extrinsic pathway, one would define it as the spark that we require to get everything running. Okay. So the extrinsic pathway produces little amounts of thrombin, but this amount of thrombin produced by the extrinsic pathway is enough to produce a positive feedback effect, which will activate factors 5, factors 11, and factor 8, which will result in a further continuation of the intrinsic pathway. So the positive feedback effect that is produced by thrombin is enough to produce sustained coagulation. All right, so the extrinsic pathway is simple there to provide an initial spark that will get things going. All right, so there is no need in nature for factor 12 to initiate clotting. Right, so when investigations are done, there is realization that the class factor is not really involved in the coagulation cascade. We need an initial spark that is provided by the extrinsic factor, then everything will be sustained by the positive feedback effects of thrombin. Right. So a brief summary of what happens. So we have this, an injury to a blood vessel. So an injury to a blood vessel will result in the following. So collagen fibers will be exposed. So contact between the blood and the collagen fibers will result in a couple of platelet reactions. So we have platelet adhesion, we have platelet activation, resulting in platelet aggregation, which ends as the formation of a platelet plaque. The blood vessel is going to contract, what we call vasoconstriction. So the muscle layer, the smooth muscle layer within the walls of the blood vessel is going to be activated and results in vasoconstriction to stop the loss of blood through the injury site. Another thing that we find is the release of tissue thromboplastin from the underlying tissues. Okay, thromboplastin is going to activate what we call the extrinsic pathway, which will result in the formation of thrombin, which produces fibrin and produces a definitive hemostatic plaque. So the fibrin meshwork together with the platelet plaque, they form what we call a blood clot or a thrombus. Okay, so this marks the end of your secondary hemostasis. Right, so there is need in many cases to prevent clotting in your normal vascular system. Remember, the adjacent portions of your blood vascular system are still intact. So if the coagulation cascade is not regulated, it means that the clotting of blood will escalate to all the other parts of the intact blood. So we need to look at the systems which are put in place in order to avoid such things happening. Okay, so the normal anticoagulation systems are divided into at least 3 mechanisms. There is at least 3 anticoagulating mechanisms that are put in place to avoid the clotting of blood. Okay, so if any one of these anticoagulant mechanisms is defective, it results in a condition called hypercoagulability. Okay, so the first mechanism in a normal person, let's see. So the first mechanism involves what we call antithrombin 3 and heparin. Okay, so that's the first mechanism. Antithrombin 3 is a plasma protein which is found within your blood plasma. Okay, so antithrombin 3 has a main function of inactivating thrombin. So antithrombin 3 will bind to thrombin and inactivate it. On top of inactivating thrombin, it also inactivates other plasma proteins like protein factor 2, 9, 10, 11, and 12. So we have these important mechanisms set in place. So we have the mechanism which uses antithrombin. We're going to have another mechanism which uses thrombomodulin. We're going to have another mechanism which uses what I call protein C. All right, so the endothelial cells which are found on the adjacent surface, like I mentioned in primary hemostasis, have the ability of releasing what are called prostaglandin. Okay, so they release prostaglandins or prostacyclin. They also release nitrogen monoxide. These two will act to prevent the formation of a clot on the parts of the blood vessels which are still intact. All right, so other natural inhibitors, they include what are called protein C. So protein C is formed in the following way. The mechanism is triggered by thrombin. So when thrombin is formed by the conversion of prothrombin into thrombin by the action of the Stewart-Power factor, thrombin will then activate protein C. Okay, so within the surface, on the surface of endothelial cells, there is a receptor that is known as thrombomodulin. Okay, so thrombomodulin will bind to thrombin. It will then inactivate all of the coagulating factors or characteristics of thrombin. So when thrombomodulin binds to thrombin, thrombin will further bind to protein C and activate protein C, right? So protein C works together with a cofactor known as protein S to inactivate the factors 8 and 5, all right? So activated factor 8 and factor 5 will be inactivated. We saw these factors being directly activated by thrombin. This time we are looking at them and seeing them being inactivated indirectly by thrombin. So thrombin acts both as a procoagulant as well as an anticoagulant. Okay, so protein C will inactivate factors 8 and 5. If you inactivate these two factors, it means that factor 10 and factor 9 will not function properly. All right, so activated protein C will also inactivate inhibitors of what are called tissue plasminogen activator. We'll talk about plasminogen activator in fibrinolysis. Okay, so this is what activated protein C does. Inactivates factor 8 and factor 5, also inactivates the inhibitors of tissue plasminogen activator. So plasminogen activator will convert a plasma protein that is called plasminogen into plasmin, also known as fibrinolysin. Okay, so the fibrinolysin will act to dissolve the formed blood clots. Okay, so another natural pathway involves antithrombin. Antithrombin is a protease inhibitor which inhibits clotting factors. We said antithrombin together with its cofactor called heparin. Heparin is a natural anticoagulant. Okay, it is a cofactor to the inhibitor which is antithrombin. So antithrombin 3 together with heparin will form a complex which will inactivate the clotting factors 2, 9, 10, 11, and 12, right? So the last, okay, so another one of the required mechanisms to prevent the clotting of blood on the peripheral or adjacent portions of the intact blood vasculature system involves what are called the tissue factor pathway inhibitor. Okay, so the tissue factor pathway is also known as the extrinsic pathway. So an inhibitor to the tissue factor or an inhibitor of thromboplastin will ensure that the tissue factor no longer produces the external spark, that spark that is required for the coagulation cascade to start functioning. Okay, so the tissue factor pathway inhibitor will inactivate factor 10 and factor 7. Factor 7 is involved in the extrinsic pathway. Factor 10 is involved in the common pathway, which serves as a link between the extrinsic and the intrinsic pathway. All right. So these two are what we have. All right. So that is all we have on the natural inhibitors. So do not forget the natural inhibitors. We mentioned protein C. We also mentioned antithrombin 3, which act together with heparin. And lastly, we mentioned the tissue factor pathway inhibitor. All right. So the following slide provides a summary of the functions of the endothelial cells. So endothelial cells have a vast amount of functionalities in the process of hemostasis, both in ensuring the formation of clots and as well as acting in the opposite direction. Okay, so endothelial cells, they normally provide a barrier between the blood and the subendothelial connective tissue, which when damaged, they'll provide a conduit between the blood and the subendothelial tissue, which then results in clot formation. Okay, so the platelet aggregation and the formation of your tissue factor, factor 7 complexes will not be triggered if the endothelial cells are still intact. Okay, the endothelial cells are also responsible for producing the anticoagulants or the inhibitors of coagulation, which are prostaglandin I2 and nitric oxide. They secrete the tissue factor pathway inhibitor. So the tissue factor pathway inhibitor is secreted from your endothelial cells. The endothelial cells are also responsible for binding thrombin, okay, via a cell surface receptor known as thrombomodulin, which then activates protein C. Protein C will inactivate the clotting factors 8 and 5. So 8 and 5, these are going to be your core factors for the factors 9 and 10 respectively. Endothelial cells will also display heparin, which acts as a core factor to the anticoagulant antithrombin 3. Okay, so heparin will elicit the activities of antithrombin 3. So it acts as a core factor to your antithrombin 3. Lastly, endothelial cells will also secrete what we call your tissue plasminogen activator. The tissue plasminogen activator is normally inhibited, okay, by a specific inhibitor. This inhibitor is inactivated by protein C. All right, remember that. So, all right, so the following list the actions or the functions of thrombin. Okay, so I mentioned earlier that thrombin acts both as a procoagulant and as an anticoagulant. Okay, so as a proagulant, thrombin will catalyze the step which converts inactive fibrinogen into active fibrin. So this is factor 1 into factor 1A. Thrombin will also activate through its positive feedback effect the following factors: factor 11, factor 8, and factor 5. It also activates the fibrin stabilizing factor known as factor 13, right? So thrombin will also activate platelets, all right? So activation of platelets involves the expression of different receptors on the cell surface membrane of platelets. So you realize that some of the steps within the coagulation cascade are carried out on the surface of platelets, and for that to occur we need receptors for the different clotting factors. These receptors are activated or are expressed on the surface of platelets by the action of thrombin as an activator. Okay, so thrombin also acts as an anticoagulant by activating protein C. So thrombin, after binding to thrombomodulin, will lose all of its coagulating properties and become an anticoagulant. It will activate protein C, which will then inactivate clotting factors 8 and 5. These two are said to be cofactors of different clotting factors. Okay, so that's what we have. Okay, so we have mentioned three major mechanisms that are classified as natural inhibitors of clotting. Okay, so we mentioned protein C, antithrombin 3, and lastly we mentioned the tissue factor pathway inhibitor. All these three pathways, they act to reduce or limit the formation of a clot on the parts of the blood vessels which are intact. The system that I'm going to describe now, known as fibrinolysis, is not involved in limiting clotting. Okay, it's involved in the dissolution of a clot. You ask yourself the question, what happens after you've formed a clot and you now want to repair the worn out vascular tissues? You obviously have to remove the blood clot. Okay, you have to remove the blood clot and lay down new tissue cells after having removed the blood clot. So the removal of an already formed blood clot is known as fibrinolysis. Right, so this is known as the thrombolytic system. So we need yet another plasma protein which remains trapped within the blood clot. This plasma protein is called plasminogen. Plasminogen is the inactive form. It is activated by what we call tissue plasminogen activator. This tissue plasminogen activator is released by your endothelial cells. All right, it was inhibited by what we call a tissue plasminogen activator inhibitor. So that inhibitor is inactivated by protein C. Then the tissue plasminogen activator will convert plasminogen into plasmin, also known as fibrinolysin, because we want to lyse the fibrin strands. Okay, so plasmin will digest the fibrin threads and other clotting factors and remove the clots. So that's essentially the process of fibrinolysis. So this here is just a summary of the fibrinolytic system. So you have your tissue plasminogen activator. It activates the conversion of plasminogen into plasmin. We also need your prekallikrein. We also need activated factor 12 prekallikrein, activated class factor. Okay, they convert plasminogen into plasmin. What factors will inhibit this? We have what we call alpha-2 antiplasmin, but we don't talk about antiplasmin right now. We don't need to talk about it. So plasmin will then activate the proteolysis, okay, of the fibrin meshwork, the fibrin threads. So fibrin will then be converted to a degraded form. All right, so the degradation products of fibrin will get them by the activity of your fibrinolysin or fibrin. So that's essentially what we call the fibrinolytic system. So next up, we have abnormalities in the process of hemostasis. Okay, so any abnormality in hemostasis will result in a disorder. So we have bleeding tendencies in the process of hemostasis. All right, so we have platelet disorders. For example, platelet disorders, they come in different forms. So we have thrombocytopenia. Penia means low. So a low count of thrombocytes, there's a low platelet count. So if you have low platelets in your blood circulation, definitely it's going to be hard for the blood to clot, right? So we have bleeding usually from many small vessels. Bleeding from vessels is known as hemophilia. What causes these? We'll look at these things. So many small hemorrhages occur through the body. We have purplish blotches, thrombocytopenia purpura. Okay, you get something called a purpura, thrombocytopenic purpura. So purpura refers to purplish blotches within your skin which are usually visible to the human eye. These occur as a result of thrombocytopenia. Your blood vasculature system hardly forms clots, so you form these purplish blotches due to small hemorrhages occurring throughout the body system. There is a picture that will be shown to show you what purpura is. So bleeding does not occur until the number of platelets in the blood is less than 50,000 platelets per microliter. So this is the threshold. Below this, you get what are called thrombocytopenia. All right. So next up, we have causes of thrombocytopenia. Thrombocytopenia may be caused by a problem or a defect with your immune system. It may be caused by the injection of heparin. Okay, so heparin injections may result in thrombocytopenia. Heparin is essentially an anticoagulant, but continuous injections of heparin may result in a low platelet count. Okay, we have quinines. If you know about quinines, we have infections that may affect your platelets. We have deficiency in nutrients. Okay, we have pregnancy and chronic alcohol abuse. All these are causes of thrombocytopenia. All right, so there you go. This is your thrombocytopenic purpura. So these purplish blotches that occur as a result of small hemorrhages along different parts of your skin. All right, so these blotches, they result from thrombocytopenia. Okay, so we also have the following abnormalities of hemostasis. So what are called defective coagulation. So now we are looking at the different defects along the two pathways. So the defective coagulations, they may be classified as being acquired or being hereditary. Acquired includes liver disease and vitamin K deficiency. The liver plays a pivotal role in the formation of blood clots. Okay, so the liver one will produce a lot of your clotting factors. There's at least 6 of them which are formed in the liver. Okay, your protein C, your protein S, protein factor 2, so the clotting factors 2 and other clotting factors are produced in the liver. They will be mentioned in some of the slides that follow, the factors that are produced in the liver. The liver also produces different bile salts which are important in the absorption of vitamin K. Okay, vitamin K is necessary for the production of different clotting factors. So vitamin K is essential for the gamma carboxylation of glutamic acid. Okay, this reaction known as carboxylation. Glutamic acid is converted to gamma carboxyglutamic acid in the presence of vitamin K. So if you have vitamin K deficiency, that vitamin K is deficient, it's not there, it's present in small amounts, then you won't have this carboxylation reaction. Hence, one will not clot normally. All right. So another thing, you have hereditary diseases, hereditary disorders. The most common is hemophilia, the love for bleeding. Okay. So one is said to love bleeding if they have an absence or deficiency in clotting factor 8. So the most common type of hemophilia present in 85% of individuals is known as hemophilia A. So you have absence of your clotting factor 8, otherwise known as anti-hemophilic factor A. So you have hemophilia A. Hemophilia B is absence of your clotting factor 9, known as the Christmas factor. So if you hear people talking about the Christmas disease, they're talking about hemophilia B, right? It's present in 15% of the population. So now, here is a broad explanation of what happens when one has a deficiency of vitamin K. Vitamin K is a fat-soluble vitamin necessary for the formation of factors 2, 7, 9 and 10. Okay, 2, 7, 9 and 10. So factor 2 is prothrombin, factor 7 is known as procalcitonin, so factor 9, then we have factor 10, the Stewart-Proer factor. So these are all produced in the liver. In addition, we also have protein C and protein S. All right, so they're all synthesized in the liver and they depend on the presence of vitamin K. All right, so I talked about the gamma carboxylation of glutamic acid that's carried out by the liver. All right, so most common cause is failure of the liver to secrete bile which is necessary for the absorption of vitamin K. So vitamin K deficiency is closely related to liver disease. We also have that. So lack of bile causes problems in digestion and fat absorption. Hemophilia, the broad explanation, hemophilia is an X-linked disease. So it means it mostly occurs in boys. Females act as carriers of the disease. Hemophilia A, due to absence or deficiency in factor VIII, is present in 85% of the population. 55% of the population, they get hemophilia B, absence of factor IX. So this is bleeding severely. So severe bleeding as a result of minor injuries most of the times. Call that hemophilia. Okay, so hemophilia sometimes may result in what are called a hematoma. So a hematoma is excessive bleeding into the interstitial spaces when blood enters your tissue spaces and shows up on the skin as this huge red patch. Okay, so we call that a hematoma. Hemophilia is excessive bleeding, but it might end up causing a hematoma. So the following is a diagram showing a huge hematoma. Okay, so this might be due to excessive bleeding, internal bleeding, what we call hemophilia. Next up, we look at anticoagulants, all right, which are used clinically. So we have different anticoagulant drugs, all right? So anticoagulative drugs. So these may be classified as in vivo or in vitro. So the ones that work within the body, we have heparin. Heparin is administered intravascularly, okay? Then we have oral drugs known as the coumarins. We have dicumarol and warfarin. So dicumarol was discovered early in the 1940s, okay? It was then superseded by the common anticoagulant known as warfarin. Okay, so both of these, they inhibit the vitamin K epoxide reductase enzyme. So they inhibit the absorption or the functionality of vitamin K. This, as a result, will prevent the gamma carboxylation of glutamic acid. Okay, so glutamate residues will not be carboxylated. Failure of this, all the clotting factors produced in the liver will not be transported into your circulatory system. So you have deficiency, you have different clotting factors. So this is how we achieve anticoagulation. All right, so another thing is we have these which work in vitro. So these are used in different experiments in petri dishes and in the laboratory. So we have heparin. Heparin is also used as an anticoagulant. We have calcium deionizing agents. Okay, so we mentioned calcium. Factor 4, all right? Factor 4 is required in many of the steps within the coagulation cascade, especially the step in which factor 9 converts factor 10 into activated factor 10. We require the presence of calcium there. So if we neutralize calcium or we precipitate the calcium within your blood plasma, it won't function as a coagulating substance, okay? This simply outlines the differences between the two major orally administered anticoagulants, heparins and the coumarins. So we talked about heparin and dicumarol. Okay, so heparin is from an animal and coumarols is from plants. Okay, we talk about dicumarol, so the parent of heparin. So the onset of action, this one is fast onset, okay, this is slow onset, takes 1 to 2 days to be activated. Okay, so this is what we call potency, the amount of time that the drug lasts in the human system. So heparin lasts for a small amount of time, up to 4 hours. This one lasts for days. This is the method of administration. This is the mechanism of action. Okay, so heparin will combine with antithrombin 3 and increase the effectiveness of antithrombin 3 in removing thrombin. So the coumarins, on the other hand, will act by competitive inhibition of vitamin K. Okay, so vitamin K is inhibited, then we inhibit the formation of factors 2, 7, 9, and 8. All right, so these ones acts in vivo, in vitro. This one only acts in vivo, the coumarins. Okay, so an antidote to heparin. So we no longer want the anticoagulating effects of heparin. We give you what I call protamine sulfate. For the coumarins, we just give you vitamin K. Okay, you'll be good to go. That's the antidote for the two. So now we talk about in vitro coagulants. We mentioned these. This is just being explained in detail. So your calcium deionization agents, so the deionizing agents of calcium, the ones that are responsible for precipitation of calcium. We have oxalate compounds. For example, you may use sodium oxalate to get your calcium oxalate. So it's toxic. We do not use these for blood transfusion. You have your citrate compounds. They combine with calcium in the blood, give it an unionized calcium compound. Okay, so it can be used in blood transfusion. These are not toxic because they produce an ionized calcium compound. Right, this is an ionized, this is an ionic compound. It produces ions which are toxic during blood transfusion. Right, so this is the different calcium deionizing agents. So just to summarize everything that we looked at today, we explained in detail what secondary hemostasis is. So we mentioned it to be the formation of a stable clot with a fibrin meshwork. Okay, so secondary hemostasis involves what I call the coagulation cascade, which was divided into the intrinsic and extrinsic pathway. It is called the intrinsic pathway because all the factors required for the pathway are found within the blood vascular system. Okay, the extrinsic pathway is so called because we need the tissue factor which is found outside the blood vascular system. Okay, so it's extrinsically located. Okay, so the two pathways we mentioned that they are controlled by the action of procoagulants and anticoagulants. Examples of procoagulants, we mentioned thrombin, we mentioned all the Thayton clotting factors. The most important ones were fibrinogen, we also mentioned thrombin, we mentioned as well prothrombin and thrombin. Okay, so the anticoagulants, we mentioned the most common one, we mentioned the fumaroles, we also mentioned heparin. Then we have the natural anticoagulants that we mentioned. We mentioned protein C, that's a protein. We also mentioned the tissue factor pathway inhibitor. Then lastly, with antithrombin 3. All right, so the other thing that has to be mentioned is synthetic anticoagulants. Okay, we have different synthetic anticoagulants and drugs which function as anticoagulants. A good example of an anticoagulative drug is aspirin. Okay, aspirin functions to inhibit what are called cyclooxygenase enzyme. We have different enzyme, COX-1 and COX-2. COX standing for cyclooxygenase. Okay, COX-1 functions to convert arachidonic acid into thromboxane in the platelets. COX-2 functions to produce prostacyclin, also known as prostaglandin I2, from arachidonic acid in the endothelial cells. Okay, so we also have what we call streptokinase. Streptokinase is essentially a primordial fibrinolytic agent. So if they ask you about streptokinase, it is a drug that is used as an anticoagulant. Aspirin is the most common one. Okay, so aspirin may be administered to patients with myocardial infarctions. Okay, so if you've had a heart attack, they might give you aspirin to prevent further blockage of your blood vessels. Right, so another one is known as urokinase. Then lastly, this is an interesting one, we have what we call a plasminogen activator. Okay, so it prevents clotting, is derived from the saliva of a bat species known as the Desmodus rotundus. So we derive this plasminogen activator from the saliva of a bat. So the full name for this plasminogen activator is called the Desmodus rotundus salivary plasminogen activator. That's what we call it. So these four are the most common anti-clotting drugs that you might be examined on. Okay, so this marks the end of the presentation. Lastly, I also have a few MCQ questions that we have to answer just to consolidate the knowledge that we have. All right. Okay, so here are the MCQ questions, right? So we'll go through all the MCQ questions and provide explanations for them quickly, all right? So I urge you to pause the video at this moment, try to answer all these questions. These are true or false questions, and come back for the explanations and see if you really understood the stuff, all right? So let's go. The following are clotting factors, okay? So fibrinogen, true. Fibrinogen is inactivated clotting factor 1. It's converted into clotting factor 1A, which is fibrin, by the action of thrombin, okay? So prothrombin, true. It's inactivated clotting factor 2, okay? So prothrombin is the one that gives us thrombin. Tissue factor, true. This is clotting factor 3, right? The one that's going to initiate the extrinsic pathway of the coagulation cascade. Calcium, very true. This is our clotting factor 4. That's calcium. Thrombomodulin, no. Thrombomodulin acts in the anticoagulation cascade, okay? So it's a cell surface receptor on the endothelial cells. Next question. The following factors are of the intrinsic pathway. So the following are factors of the intrinsic pathway. The tissue factor, false. It's for the extrinsic pathway. Factor 7, false. Extrinsic pathway. Factor 12, true. That's the class factor. The first factor that we have in the intrinsic pathway. Factor 9, true. Works together with factor 8 to activate the Stewart power factor, which is factor 10. Okay, so lastly, we have factor 8. Definitely, that's the cofactor to factor 9. It's a clotting factor for the intrinsic pathway. Next question. The following are factors that oppose clot formation. Okay, so protein C, one of the mechanisms of the natural inhibitions to clot formation involve protein C. Okay, protein C bound to thrombin, which is bound to thrombomodulin. Okay, protein C will then inactivate proteins 8 and 5. For the protein factor 5 and 8 are inhibited by protein C. So it's there in the opposition of clot formation. Factor 7, no, factor 7 doesn't oppose clotting, so that's false. Tissue factor pathway inhibitor, true. That's another pathway. So we mentioned that the endothelial cells will release what we call the tissue factor pathway inhibitor, which is going to inhibit the extrinsic pathway. We have thrombomodulin, true. We have antithrombin 3, true as well, right? So antithrombin 3 works together with heparin to inhibit some of the clotting factors, right? So another question says the following are anti-clotting drugs, right? Interesting. Aspirin, we mentioned that. So this inhibits the COX enzymes that convert your arachidonic acid into prostaglandins and thromboxane. Okay, so recombinant tissue plasminogen activator. Yes, this is a therapeutic drug that's given to people without your tissue plasminogen activator. So people with certain hemostatic disorders are given recombinant tissue plasminogen activator. So it's a drug. Streptokinase, true, we mentioned it. So Desmodus rotundus salivary plasminogen activator, that's also another drug, an anticoagulant, anti-clotting. Heparin, this is also true. Yes, it's true. It's an anti-clotting drug. So it's given to people both intravenously. It's given to people intravenously. Yes, it's found naturally within your blood system. So it's going to be natural and both synthetic as well. So concerning hemostasis, the mechanism is most effective in injured small vessels like capillaries. Okay, so this is true. If you injure major blood vessels like an artery, okay, hemostasis will not be functional in preventing the oozing out of blood. Okay, so you need surgery in that case. So this is where vascular surgeries come into play. Okay, so Hemostasis is only good for small blood vessels like capillaries and arterioles. Okay, so accumulation of blood in tissues as a result of a bleeding vessel is called a hematoma. True, we saw the picture of a hematoma which resulted from hemophilia. Okay, so the von Willebrand factor is involved. True, it's involved in hemostasis, specifically the primary hemostasis. Okay, so it's involved in adhesion of your platelets to the underlying collagen fibers. Thrombin is not important in hemostasis. This is a lie. We said thrombin lies at a central position in the entire process of hemostasis. Okay, it is a major factor. If you want to forget about all the factors, please remember thrombin. Okay, so lack of factor 8 causes hemophilia. True, 85% of people have hemophilia A. This is easy stuff, all right? So, what's next? So, this question involves most of the stuff that we didn't do today, but if you watched previous segments from the channel, you might be able to attempt these questions, but I'll do well to explain most of the things. It seems like an interesting question. Okay, so the first one says people with heterozygous sickle cell traits are resistant to malaria. So the answer is true. Okay, so people with the sickle cell gene, heterozygous recessive, the sickle cell trait tends to protect them against malaria. So when one gets the malaria pathogen, okay, it has to inject itself into the red blood cells, where it then further matures and reproduces. Okay, so the sickle cell red cell provides less conducive environments for this process. Okay, so the malaria pathogen finds it hard to mature inside a sickle cell red blood cell. So people with this sickle cell trait have been recognized to thrive with the malaria pathogen. They're not really affected, they survive. Okay, so this is true. Folic acid is not required for the synthesis of erythrocytes. This is false. Okay, so we need folic acid in part of the steps involved in erythropoiesis. All right, so next question. Serum is plasma with no clotting factors. Keyword on no. No clotting factors. So this is false. It has a bit of truth to it. Okay. So serum is plasma with removal of only 4 clotting factors. Okay, so we have fibrinogen, we have clotting factor 2, clotting factor 5, and lastly clotting factor 8. That's what serum is. So it still has a remainder of clotting factors within it. Okay, so D, pale skin can be a symptom of anemia. This is true, specifically iron deficiency anemia. Okay, so it's the most common type of anemia which has a symptom of pale skin. So estimated sedimentation rate is increased during inflammation. I suppose this is a typo. It was supposed to be erythrocyte sedimentation rate. Okay, so this talks about the rate at which red blood cells sink or sediment. Okay, so under normal conditions, red blood cells are very light, so they easily float instead of sink. They take a long time to sediment. But in cases of inflammation, red blood cells, they aggregate into clumps. Okay, so you find clumps of erythrocytes in inflammation. So an inflamed person has clumps of erythrocytes. So a clump has an increased weight than a single red blood cell. So clumps of erythrocyte, they easily sink to the bottom. So the erythrocyte sedimentation rate is increased. So this is true. So this marks the end of our segment, folks. This has been quite a pleasure. Hopefully you got to understand all the intricacies of the complex pathways of hemostasis. Okay, so if there's anything you feel wasn't touched in this segment, feel free to get in touch with us. We'll leave the contact details on the comment section. If there is other things that you would like for us to touch on in the next videos, so mention your comment in the comment section. Otherwise, as always, stay positive, keep looking out. Ciao.